You'll note that the cerebellum, just indicated there, appears to be connected to the back of R1. It's indeed the hemispheres of the cerebellum do develop from R1, but Lewis has suspected from the chick work, and we've now shown in the mammal, that the vermis of the cerebellum comes from the isthmus. So the isthmus gives rise to the vermis, the first rhombomer to the hemisphere of the cerebellum. There may be a tiny contribution from R2, but it's not convincing yet. It may generate the flocculonodular lobe. It seems a possibility. But as we go further forwards now into the remainder of the telencephalon, we've got a further series of subdivisions and some other things to note. And I suppose the most important one first is that the preoptic area situated rostral to the hypothalamus is not a hypothalamic structure. It's actually a separate area. There's a wall dividing the two gene expression territories between those, and from this point onwards we start to divide the remaining part of the telencephalon into the pallial and subpallial zones, which makes for a further story. And which I've just summarised here because I'm not going to take you through more pictures to try and show those. In level 6, we divide the telencephalon into the pallial and subpallial domains, and the pallial subdivisions probably are familiar to you: the medial, dorsal, lateral, and ventral. The medial being the hippocampal, the hippocampal structures, and the dorsal essentially being the neocortical. But the subpallial divisions are the striatum, the pallidum, the septum, and preoptic. And this new word that Lewis has introduced, the diagonal domain. It's what you'd be familiar with as the horizontal and vertical nuclei of the diagonal band, and the magnocellular preoptic, which actually turns out isn't preoptic, but that's always a problem we have with this, where people have named things for one area and then it turns out they actually belong to another territory. It'll take a little distance to do that. And then, of course, at level 8, we have some complications, because the amygdala in particular doesn't just derive from one of these domains. It has cortical elements in it, it has striatal elements, and it has ventral olfactory cortical elements within it. And so it becomes rather harder to represent in the ontological structure from that point through. And but nonetheless, this is essentially the picture that we get of how you apply this to the mature brain, and to see the various parts of that. For those of you who are interested in particular areas and you want to see what has been done to your area with this, you need to have a look at the detailed hierarchy within those areas to actually see exactly what's been shuffled around and what you might have to take into account, or you might want to go back to your own version of that. Which does it. And at the moment, as I said, the full 6-level ontology is on the BrainNavigator site, and the importance of that is that the BrainNavigator site is able to show you equivalent structures in mouse and rat and monkey and now human. We're just going to move the human across to it in the next step with the acquiescence of Jürgen May, to make sure that, so you can click on a structure and you can see the ontological position that it holds, and then you can see the same structure in these other species, essentially being homologous structures as you go forward with that. Of course, one important thing here is the presence of the optic nerve coming from the floor plate region just there, indicating that we're going out towards the eye. I'm not going to try and explain further the other subdivisions that Lewis has done for these, but say that the last 3 levels, 11 to 13, are the things which you actually find in the mature brain and find in individual structures. It's interesting when people ask about the Rat Brain Atlas, they occasionally, in what I think is perhaps a naive way, alert you to things which you hadn't thought of before. And a few years ago, and I've often discussed this with George, someone accosted me and said, look, the Atlas is okay, I think, but the new version of it doesn't have the superior colliculus in it. And I thought, oh no, hold on a minute. Like, it does have the supercolliculus, it has all the layers and everything else. They said, doesn't have the word. I couldn't find superior colliculus anywhere. And of course there's a truth in that, and that is that these supraordinal terms must be available somehow or another in the system, and that's fortunately what the BrainNavigator system does, is that you can go to superior colliculus and it will take you down to the other parts of it within there. So you don't have to be an expert in the superficial, grey stratum or those sorts of things in order to. You can actually use the word superior colliculus, because most people who use atlases actually want to find the hippocampus or the superior colliculus or something pretty big. They're not actually interested in the fine detail that we might have been from the beginning. So it does actually alert you to the fact that we needed to do something different. It was then we really started thinking about what we had to do with that. So, as I've said, we're for the moment resisting Lewis's move to the 13 layers, and I think it's very important that that is published on its own, because it contains the most consistent relationship between the gene expression data as they go forwards and the names that we actually have. But I think that we'll either end up with 6 or maybe 7 if we have to get subdivisions of nuclei at the bottom end in order to accommodate the needs of ordinary people, so that means we have to collapse some of the layers that Lewis has had in his 13-level ontology, and it is, I think, suitable for adult nervous system. I put this together with some difficulty, and I realised as I did it that there's no way I could sell this to you as a way of understanding it, but in your notes you'll have it. If you're interested in how Lewis's 13-level ontology collapses into 6, this is an attempt to explain what the relationships of that are. But if I tried to do it, I'd probably fumble, and it would probably take a half an hour, so we're not going to do that. I do want to show you what it looks like in BrainNavigator, though, because if you're looking for parts of the striatum, for example, you'll find out that the caudate putamen or the amygdalostriatal transition zone are part of corpus striatum, which are part of the striatal area as broadly defined, which is part of subpallium, which together with pallium forms the telencephalon, and there from the prosencephalic vesicle. So you can, with any structure, as you open it in BrainNavigator, you can see the parents going straight back, and you can also see the other things that exist at the same level as it, so that when you find striatum and you drop down, you find that the other subpallial structures are pallidum, diagonal domain, subpallial amygdala, subpallial septum, and preoptic area. So you can see the things which fit into the same level in the ontology, which gives you a good picture of that. And as I said, what's particularly nice is that whether you care about it or not, whenever you click on any structure or open a section and identify it, the ontology will appear on the left-hand side and show you the parents and show you the other relationships. And I think this was just this is the human brain. The human brain pictures are now on BrainNavigator, but because that's still in the traditional form, this gives you an idea of how the traditional ontology will divide things up, and it won't be the same. We're hoping to make that conversion over the next 6 months or so to make sure that you can read that in the same way as you could read the rat or the rhesus monkey. And there are a few outstanding changes which people will have to adapt to if they choose to go the way of this ontology. And the first thing is this term diencephalon. Those of you who did standard medical school anatomy know that the diencephalon has thalamus, epithalamus, subthalamus, and hypothalamus as parts of that. And this doesn't allow that. First of all, it says that the diencephalon is the 3 prosomeres, and it doesn't include the hypothalamus. We also find that the subthalamic nucleus pops up in the hypothalamus, and the epithalamus disappears as an entity because it's simply part of prosomere 2, sits on the top of the thalamus. As I've said before, we find the hindbrains divided into 12 areas, the 11 rhombometers and the isthmus. The cerebellar derivation is different to what people thought, and as I pointed out, it's really interesting when you look at a structure like the substantia nigra. Because of the feet of each of those prosomeres, it has to actually extend right across those as it curls around from isthmus up to P3. And you'll be pleased to know I'm just about to stop. This is a question which George is going to address now in part. I'm afraid it's something we do feel pretty passionately about, and that is that people, when they're publishing neuroanatomical papers, always have a choice of what abbreviations will they use. If they've got the lateral geniculate nucleus, will you call it LG, LGN, DLG? You know, there's a variety of things you could call anything, and in fact, probably half of the people or more just make them up. And this creates a terrible problem. I mean, whenever I read these papers, I think, what on earth does this mean? Because I look at some abbreviation that someone just had a great idea that afternoon and they thought, oh, we'll just call it this. And so no gene journal would ever allow you to do that. If you've got EGR2, you've got to put down EGR2. I mean, maybe you get away with CROX20 because it was older, but they basically you have a canonical set and you have to stick with that. Swanson's abbreviation set is not too dissimilar from ours, and it is an important atlas because, you know, Larry's one of the great forebrain anatomists, and but simply because of the length of time our atlas has been out there, we're up to 50,000 citations. They weren't all congratulating us on the abbreviations. They're mostly using the coordinates, but it does mean that a lot of people have looked at it, and many people do use it. And so we really believe that if you're going to use an abbreviation set, use either the Swanson set or our set, and use some identified set, and then everyone knows where you're going with it. We think that's a better way to do it.